So it's my distinct pleasure to introduce the speakers for the first speakers for this year 2021 MnRI series. So this is these are doctors Aaron Lorenz and Suma Sreekanta. Aaron received, he's an associate professor in CFANS and received his PhD from University of Wisconsin in 2008. Aaron, am I right?
Yes, that's correct.
If my memory. And he joined the University of Minnesota in 2015. And Dr. Sreekanta received her BS from the University of Miami and her PhD from the University of Minnesota. And the title of the talk is Developing Phenotyping Solutions Towards Optimizing Soybean Shoot Architecture. So without further ado, the podium is yours. Thank you again for doing this presentation for us.
Yeah, you're welcome. Thanks for the opportunity here, Nikos, and thanks for the introduction. So the title is already stated here, so I think I'll just jump right into it. But a little bit of a bigger picture here is we want to introduce all of you out there to some of the things that we're doing and some of the questions we have in terms of improving soybeans and making them even better, and trying to basically find some common areas of interest and potential for collaboration where we can put our skill sets and questions together to maybe do some great things. And so kind of keep that in the back of your mind as we go through this presentation. So if I can change. So yeah, I guess as a start off here with a typo in my first line of my outline. Soybeans, I'm gonna start off with just kind of talking about soybeans and what they are. I don't really know who my audience is out there too much, and I'm gonna assume that you know very little bit about plant science, you know very little bit about agriculture. Maybe you don't even know what soybeans are. Maybe you are a new student here and haven't had a chance to drive around the Midwest very much lately because of the pandemic, for example. So I'm gonna go through what soybeans actually are. Then I'm gonna talk about the concept of crop canopy and how crop canopies can be optimized to improve yield potential in a crop like soybean as well as other crops. And then I'm talking about some of the challenges we have related to genetically altering these crop canopies to make them even better than they are to maximize yield potential. All right, so Suma will kind of go into that, of how we're looking at the shoot architectural properties of individual plants, how those come together to create the crop canopy, and how those may be genetically changed to enhance yield potential if only we could measure those properties more efficiently. And then we'll go into some future goals and vision we have for this kind of research. So to kick it off, yeah, these are soybeans. Soybeans, the scientific name is glycine max. You know, if you go down the road, they're basically the short ones out there. So you see the tall ones are normally corn and the short ones are normally soybeans. And so if you haven't lived here very long and you get around to Minnesota and the rest of the Midwest, you'll see a lot of soybeans. Just in terms of some statistics, soybeans are a very widely grown crop. They're on over 83 million acres in the United States. You can see the distribution of those acres here in this map, pretty much as you'd expect, concentrated in the Midwest, a little bit in the Mississippi Delta region down here, a little bit out east. One thing about soybeans that's been kind of interesting is that they've migrated northward pretty steadily over the last several decades. So it wasn't that long ago, maybe 20 years ago, 30 years ago, there weren't really many soybeans up in North Dakota and northwestern Minnesota. And soybeans have gradually marched northward, and actually soybeans are now marching northward up into Manitoba. And a lot of that northward march is attributed to, well, of course, climate change, but also just selective breeding, developing earlier and earlier maturing soybean varieties that are adapted to those northerly conditions. Okay, so additional crop opportunities for farmers. So, you know, once again, in Minnesota, we have over 7.4 million acres of soybean. It's the second most widely grown plant crop in Minnesota. And it brings in roughly $4 billion in value to Minnesota every year. And a lot of that is exported. So it's money coming into the state. And that number fluctuates widely. Not that long ago, it was only about 3 billion because the crop prices have suddenly spiked. And so now that number is closer to 5 or 6 billion. So big numbers here. So in terms of what we use soybeans for here in the 21st century, most soybeans are processed at a refinery, what we call kind of a soybean crushing plant. And they're basically turned into protein meal, which is largely used for a protein source for monogastric livestock. And then they're also turned into oil. So about 40% of the soybean is meal, protein meal, and about 20 or so percent is oil. In the oil, the use of the oil ranges from anything from frying oils, cooking oils, salad dressings, shortenings, all that kind of stuff. Some industrial lubricants, and also biodiesel. All right, the demand for biodiesel is actually going up over time, and there's expected to be a great demand for soybean oil coming down the road here. The soybeans are also used for a lot of other things. They're used for a lot of traditional sources of protein, traditional foods such as edamame. You may enjoy edamame. Of course, they make up soy sauce. Tofu is a popular use of soybeans. We have a lot of tofu in my household. I enjoy tofu very much. And then I'm not sure if you recognize this, but this is a soybean food product called natto. It's a traditional dish in Japan, and sometimes it's eaten sort of like oatmeal. So we have in our own soybean breeding program that I manage here on campus, we have a lot of varieties that have been bred specially for this special dish called natto production. And those are put in containers here in Minnesota, and they're shipped over to places like Japan. And then, more recently, we have a lot of meat substitute products out there, meat alternatives, I should say. And in my opinion, some of the better ones on the market are actually made out of soybean. So the Impossible Whopper is made out of soybean, and the interest in these types of alternative meat products is growing quite rapidly over time. And in terms of vegetable protein, soybean is actually the most superior vegetable protein out there in terms of its overall amino acid balance. So it's good for that as well. So lots of uses of soybeans. So thinking about where soybeans actually came from, soybeans' wild ancestor is a plant that grows in a sort of prostrate manner, a viney manner, found throughout China and Korea and Russia, and it's known by the scientific name of glycine sojae. It's soybeans' wild ancestor. You can see a picture of it here on the ground. It just kind of lays in the ground. And the seeds of soybeans are very of glycine soja, this wild ancestor of soybean, are very, very small and black in contrast to what domesticated soybean looks like. So remarkable changes here that ancient farmers have made just through basic selection of soybean throughout the years, from going from wild soybean to domesticated soybean through that domestication process. And so, in addition to those nice yellow seeds and nice upright plants, other things we expect of soybeans in the 21st century, made possible by the science of genetics, are things like resistance to various pests and diseases like soybean cyst nematode, and resistance to Phytophthora, which is a fungus that affects the soybean plants. Okay, we have genetic sources of resistance for these types of things. We actually expect our soybeans to be resistant to certain classes of hybrids that have been made possible by genetics. We expect them to have very high yield, which I'll show you in a minute here. We expect them to be tolerant to things like iron deficiency chlorosis, which is an abiotic stress. We also have ways of modifying their fatty acid profile so that they have a certain type of oil composition that makes them very desirable for certain types of applications. So, and then we also expect them to be broadly adapted across a wide ranging conditions. So through selective breeding and through proper management, we expect a lot of soybeans of these days in the 21st century compared to what our ancestors would have expected of them. And just in terms of overall improvements in yield, through the power of selective plant breeding that practitioners have been carrying out now for decades, we've made some pretty considerable gains in seed yield over the time. So if you go back to, this data here is from a common garden experiment performed a number of years ago. Basically in a common garden experiment, you have varieties that are grown in the same location, same environments, basically in the same field, all right next to each other in the proper experimental design. And you can see that when you put varieties that have been developed and released in the same experiment over the period of decades, you can see the great improvement in yield here. So these varieties would have been developed and grown by farmers back in the 1920s and 1930s and 1940s. Compare their yield to what we'd expect from a variety that's been bred and grown by farmers now only in the 2000s. So you expect there's about a 60% yield increase here. So basically for every bushel of soybeans you would get from an old variety, you get a bushel and a half from a new variety. And that allows farmers to basically produce more on a unit area of land than they could decades ago. So more production just through the developing and growing of better cultivars, better varieties of soybeans. All right, so that's a little bit about soybeans. I want to talk a little bit about what we call a crop canopy. And kind of the simplest way I can put that is, a crop canopy is basically a field of solar panels for plants. All right, so if we think about an individual leaf here, all right, think about that as a solar panel that the plant uses to collect energy in the form of sunlight and takes that energy to basically power photosynthesis to allow the plant to fix carbon dioxide in the air into structural carbohydrates that help the plant develop and grow. Okay, that's what an individual leaf does. And when we put those leaves together, put those plants together in a stand of plants, that's what those leaves and stems come together to form a crop canopy. And basically, this crop canopy is an apparatus for collecting sunlight for producing food at the level of a field. And the thing about it is that the net productivity of a canopy, the net productivity of a stand of plants depends on the structural properties of its canopy, okay? And the structural properties of its canopy depend on individual plants that make up that canopy, and it depends on things such as the canopy, the age of the plants, the angle of the plants, the density of the plants, and so forth that develop that canopy and give it its properties to maximize overall productivity across an area of land. And so ideally, when plants are fully developed, across an entire field, of course, the crop canopy would cover the entire land so that no sunlight here is wasted and hitting the ground, and it's all being captured and being converted into structural carbohydrates that eventually would give rise to the plants that eventually give rise to the food that we want to harvest from that land. So the thing to remember here is that and I guess what I just said, now it sort of goes without saying that not all crop canopies are built the same way. They're not all common, they're not all the same, even within the same plant species. All right, so let me talk about why that is and why that matters. So if you have two different plants here, for example, let's call it plant X and plant Y. Plant X here on the left has what we call a very horizontal canopy. Okay, the leaves in this plant are very horizontal, as opposed to plant Y, where we have different angles of the leaves throughout the different parts of the plant. All right, so these leaves are more angled. In plant X, you can see that all the sunlight, all right, is hitting that top leaf, okay? In contrast, in plant Y, the sunlight is more uniformly distributed throughout this plant, and it's hitting different leaves up and down this main stem here. And the reason why that matters is because well, let me kind of skip this slide. This graph here kind of shows how if you look across the planter in terms of the accumulated leaf area, in plant X here, basically you're capturing all the photosynthetic photon flux density up here in the one leaf. When you get down to the other areas of the plant, leaf areas of the plant, basically you have less photons being captured, all right? Whereas in plant Y, this basically shows that the PPFD is more uniformly distributed throughout this plant, okay? So why that matters is because there's a rate that there are great diminishing returns in terms of overall photosynthesis rate with the concentration of the photosynthetic photon flux density, all right? And you can see that in this graph. So basically what happens is that leaves easily become oversaturated with sunlight. So in a situation like this, you have a leaf up here that's capturing all the light, leaving these leaves shaded. This leaf is being oversaturated while these leaves are not being utilized. All right, so you can see that relationship here, where as you move towards increasing PPFD, you have diminishing returns in the rate at which the plants are fixing carbon dioxide. And then the other part about this figure is that the circles here with the numbers in them represent the leaves, in this case, below the line for plant Y up here. These circles and these numbers represent plant X. So you can see that plant Y any individual leaf is not getting as much sunlight for plant Y, all right, but they're all about on this part of the curve, kind of getting to that asymptote for CO2 assimilation rate. In contrast, for plant X, above the curve here, you can see that the leaf 1 up here is getting a lot of sunlight, but leaves 2 and 3 are being shaded, and so they're not getting enough sunlight. Okay, so they're not maximizing their overall photosynthesis. And the net result of all that is that the overall photosynthesis of plant Y, if you do the modeling, is actually greater than it would be for plant X, again, attributed to the more uniform distribution of light throughout that plant. So this is important because we've actually seen in a lot of different crop species, just through the power of selection of breeders breeding for higher grain yield, we've seen these expected changes in plant morphology. So the classical example is in maize. If you look at old varieties of maize, okay, versus newer varieties of maize, older varieties tend to have a more horizontal leaf structure and a more horizontal canopy. In contrast, newer varieties, which have greater yield, tend to have more upright leaves. And what this allows them to do is it allows farmers to plant more plants closer together in the field and still get that light distribution throughout the whole canopy, so the plants are being efficiently used in terms of their sunlight capture. And that's really been the case in maize, where most of the gains in corn yield or maize yield have been made through the ability of planting plants, more plants more closely together, and those plants being able to tolerate those high density planting conditions as opposed to older variety of maize. This is a similar situation in rice. There's also a similar situation in wheat. Okay, so we've seen these expected changes in canopy structure and likewise shoot architecture in these plants. In soybean, on the other hand, again, once also a very important crop, has not really seen as much of this change in canopy structure, and at least we haven't measured this change in canopy structure as much as we have in other types of crops. So in a typical soybean field, you see a situation like this. You see a pretty flat canopy structure here, okay, where there's not a lot of transmission of light into the lower parts of the canopy. Most of the light is being captured on the upper leaves of that canopy, leaving, and then the leaves on the lower parts of the canopy, way down here, tend to be shaded to a great degree. So that's what typical soybeans look like. Now, what we may want, and what we really don't have yet, except in this one genotype which has this unique trait, is we may want a more vertical canopy structure. All right, in this particular soybean genotype, this has a modified petiole with a kind of a steeper angle and shorter petioles on these plants. And so you can see that they're a little more vertical, and perhaps a soybean variety like this would basically allow more light transmission into the lower parts of the canopy and get closer to that ideal archetype that I just talked about. All right, so that's kind of to summarize all that. What we want then at the end of the day is basically we want plants that are going to rapidly close their canopy so that they maximize light capture as quickly as possible after planting and not waste any sunlight. And once again, we want plants that are going to be kind of more vertical in their canopy structure to allow more light to be more efficiently used at lower levels of that plant canopy, of the crop canopy. And so with these questions and goals kind of in mind, a project that we have had recently funded by the USDA National Institute for Food and Agriculture program, their AFRI program is what it's called, as we call it a shoot architecture project. And we basically have 3 objectives related to this project. One is to characterize the genetic variation for shoot architecture traits among what we know to be 50 architecturally diverse soybean accessions, and to determine which of those types of architectural forms transmit light the best and the overall are overall better for their overall photosynthetic efficiency. And then we want to also look at different soybean cultivars that have been bred throughout the different decades and evaluate any changes in shoot architecture properties and therefore consequent canopy structure changes that could have been made through breeding for simply seed yield. And then we also want to, we have a certain gene that we know something about that seems to affect branch angle, and we want to see if that branch angle gene does indeed have an effect on light transmittance into the canopy, as well as overall yield, as well as canopy coverage. So that's kind of a specific project we're thinking about, or that we're currently undertaking. And Suma is going to talk a little more in detail about some of the challenges we have with that project in a bit. But for the time being, I kind of want to just kind of back out here, zoom out, and talk about this at a little bit of a bigger picture, and just point out that you guys may have heard this many times before about this trend of decreasing cost of genotype data. So DNA sequencing costs have plummeted over the years, and this is an important trend and has great ramifications for how we approach plant breeding in this century. At the same time, the collection of the actual measurements on the plants, what we call the phenotypic data, the cost of those has pretty much been pretty static, if not, increasing in cost. And so there's this difference between these two types of data in terms of their cost and ease of collection. And so again, the genomics revolution, if you talk about plummeting costs in DNA sequencing over time, this only goes to 2012, it's gone down even further. And if you look at databases that house DNA sequence information, along with this cost, the size of those databases holding DNA information has just completely exploded. Okay, this scale here is on the millions of base pairs that are being deposited into these databases. And so we have this massive trend in collecting DNA data. But it's best that such a revolution is also matched with a revolution in terms of our ability to measure certain features on plants. And I call that, or everybody in my field kind of calls that, sort of a phenomenous revolution. Okay, basically taking what we can to characterize the phenotype of a plant as well and as accurate and as high a resolution as we possibly can. And I have this picture of one of our fields. Whoops, go back. I have this picture of one of our fields on this slide, just to sort of give you a feeling for the scale of our trials and the scale of the phenotyping that we're trying to accomplish in some cases. And so this field is a field over by Rosemont, Minnesota, and each one of these little short rows in this entire field is actually a unique genotype. There's up to about 15 or so thousand unique genotypes represented in this single field. All right, and so we can measure this many genotypes for pretty crude traits like seed yield and plant height. But what we can't do is we can't make very detailed measurements on certain shoot architectural properties that we may want to. And it's also conceivable that we could genotype every unique variety in this field. Okay, it'd be very costly and it wouldn't be easy and be a lot of work, but it is actually conceivable that we could do that. But it's not, again, within the realm of our current possibility of using any sort of manual labor to measure the types of things that we'd like to measure on a population like this, in terms of getting a better handle on the genetics underlying shoot architecture, which Suma is now going to talk to you about in a little more detail. So I'm going to hand it over to Suma now.
Thanks, Aaron. So you guys heard what Aaron just talked about, and he showed you this huge fields of soybean plants. But my job is to kind of look a little more closely, because the ultimate unit of that entire huge field that we saw is just one plant, right? So what architecture that plant shows affects, when you have that many, affects the whole canopy. So I want to talk about a little bit about what each plant then is made up of, and each plant is made up of a single unit. It is called a phytomer, which is essentially just the leaf, the internode and node. Simple, right? But plants are amazingly plastic, which means they can grow up to be like trees or like little tiny plants. So each phytomer can then be repeated multiple times, have branches, and each branch is then like a small plant. So it builds with multiple phytomers like this. So it's just a simple unit, but it's built on one on top of the other. And so when you have something, a unit like this, you can imagine it can go anywhere because it faces environmental challenges. So plants are very plastic, which means they can change. So they might go a certain way, branch in one direction or another direction. So they're very plastic, but they also are genetically controlled, which means that a soybean plant looks like a soybean plant and not a banana plant. So they have certain basic rules on how they can build these phytomer units into larger plants. So there is that constraint, but they are also responding to environmental challenges, which means they are plastic too. Okay, so think about it like a car. As you're driving, you might come across challenges. There might be a car that's coming from a left lane, so you might want to make slight adjustments. So something like that. So plants also make those changes. And so once you have this sort of a unit, then this is a cartoon I'm showing, but you can see, you want to go to next slide? So yeah. And so here's an actual plant, which is soybean. So you can see there is a main stem, and then you have additional things apart from those main phytomer I showed you. There are other things that could be added on this, like a flower or pods or things like that. But the basic unit still remains the same. Next. So so to define it, so what we've been talking about shoot architecture. To define it, so it's essentially the overall size of the plant, the shape of the plant, the orientation, the distribution of these little units that I talk about, the simple units, how they are placed to form the main unit. That is that frame that makes the overall shoot architecture. So and like I was saying, this architecture determines how light is let in, even in a single plant, but also when you put them as canopies, and that would be like a canopy property. So how light penetrates the canopy is dependent on the shoot architecture of the plants. The next slide, please. So, Aaron showed you this huge fields and fields of these canopies, and they look pretty uniform. But in fact, what you see mostly when you go out in the fields is one variety that's grown because it's high yielding. But in soybean, there is a huge variation in shoot architecture, and this I just wanted to show you a snapshot of some of the ones that we're growing out right now. So this is essentially, you can see that the leaves look different, the stature looks different, number of branches look different, how the leaves are placed on the plant is different. So there's a lot of variation for the architectural units on the plant. So next slide, please. And in many plants, you may be able to see what the entire plant looks like, but in soybean, no such luck, because the whole plant is usually covered with leaves and we don't know what the skeleton of the plants look like. So this is defoliated plants, which means we have manually removed the leaves, so you can see what the skeleton looks like. And so these are just the branches in the main stem of the plant, and you can immediately appreciate how different they are. So each of these is a different variety. That means they are genetically distinct. So you can see that some of them are kind of like narrow and long, some of them are much broader. And so the question that Alan posed of like, how do you get good light penetration into the canopy? So we know what we think we want a plant to look like, but the question is, how do we get there? What is it that we need to change in order to get to that point? In the next slide, please. And the other question was, we talked about light interception in soybean. So this is, we've kind of tried to measure light, and believe me, it's not easy to measure light. So this is a simple instrument here. It's a Leica line sensor, which is a meter long instrument, and we kind of place it at 10 centimeter increments from the ground up to the canopy, and it measured, kind of averages the light that is incident at that point over that meter long area. Okay, and so it gives us an average of what the light quantity looks like at those points. If you want to go back, and so and in on the right, you can see I've put the variety. There are 4 different varieties, and you can immediately see the amount of light that hits at every 10 centimeter increment is vastly different for each genotype. this is mostly because of the architecture of the plant. So you can see like in the top left-hand corner of the graphs, almost up to 50% height of the plant, there is no light penetration. That means it's extremely dense at the top. Whereas the one right below that, you can see that there is a lot of light penetration even at the ground level, which means there is not as much crowding at the top. So shoot architecture actually has an impact on how light is intercepted. And this we're just talking about the quantity and not the quality of light, which is another question that we're interested in, but I won't talk about it here right now. Next, please. So where is the variation coming from? So we talked about what we want, but how do we get there? Let's say you want a plant that is kind of V-shaped, okay? Something that will let in light from the top to almost towards the bottom. But how do we accomplish that? So there is a lot of things that can change the plant structure. Like I said, it could be so here I've just shown you for the cartoon's sake, just the main stem and leaves. So it could be, for example, number of leaves, how the leaves are placed on the plant, the geometry of the leaf, the size of the leaf, the shape of the leaves, and even the leaves can move. So over a period of the day, it tracks the sun. So how does it move? To what extent it moves? All of it makes a difference. One more click, please. And so this again is a cartoon of a single plant, how the red line shows the light transmittance. So you can see top left hand corner, the light may transmit to some extent. The one next to it, because there are too many leaves, maybe not as much. The one at the bottom, maybe to some extent it lets in a lot more light, because it's kind of, the angle of the leaf is more acute. So it these kind of traits or features actually make a difference. Next. And so this again, from single plant to what Aaron was showing. So he showed you one similar to this. So this is different varieties that we're growing. And so you can see in the first one, the light kind of hits the ground. You can see it all the way towards the ground. But we don't want our architecture like that, because we don't want ground exposure for weeds to grow through. We want to utilize as much light as possible. You can see in the next one, kind of like two peaks sort of thing. So that's somewhat like an ideal architecture we want. So so each of these have distinct architecture. And so in the next slide. So our aim of the study is, while we have a generic understanding of what we want to know how to get there. So our aim of the study has been first to estimate variation in light environment. Although we've been able to look at average light at a spot, it doesn't tell us how the light scatters within the canopy. So if you like, what light environment does a single leaf placed at any point in the canopy, what environment does it face? So we don't know the answer to that question. So we want to do that more in a better way. And then the other question we want to know is, which of these shoot architecture traits impact light penetration the most? So like I said, it could be the leaf angle, it could be how many branches are there, how many leaves does each branch carry, the angle of the branches. So there are many features, or what we like to call as traits. And that's an important word. So which of these traits impact light penetration most? Is there one single trait? Is there multiple traits? Are there a combination of traits that can give us a better view of this? So, and how do we study them? So to do this, we've kind of come up with, so if you want to go to the next slide. So we came up with a 2D image processing, imaging and image processing stuff. So I want to talk about this because, Aaron was showing this huge field of soybeans, but this study is small, because manually what you can accomplish is quite small. So what we've done is we picked about 40 genotypes. So we want to get a baseline idea of what a soybean plant looks like. What are these traits and how can we measure them? So we could only, with what resources we have, we kind of decided to do like a small pilot study with about 40 varieties or genotypes. So these are architecturally very different or distinct. And so we want to ask, why are they different from each other? What features or traits make them different from each other? So this is we decided to do us like a small study. And so this was essentially by using destructive sampling, which means you cut off the plant in the field, and you either image in the field or bring it to the lab and study it. And so I've shown you some of the features or traits that we're interested in, like the height of the plant, like the what I call how far up the plant do branches exist. Then that's called the branching zone. And then leaf features, angle of branching, angle of one branch to the next branch, so things like this. So we image these. One thing though is that this is very time consuming. So you can imagine actually to harvest these plants and then kind of pull off the leaves and then uh, image them just so is extremely time consuming and labor intensive. um, and you cannot, in this way you cannot do all traits that you're interested in. It is this sort of imaging is only good for capturing some traits but not all traits. Next slide please. So by this, so what kind of variation can we see? And so you can see in the first panel, it's the petiole slope. So if you remember our previous slides, we showed you at the very top of the plant, some of them are more rounded on top, some of them are more tapered. So the how the petioles are placed. Petioles are the things that join the leaf to the main stem. So how they, the slope of these is actually important. So you can see that some of them, the slope is pretty small, but some of them are very steep. So that matters. And there's variation for angle of branching, for leaf dimension. So there are all these features, and we can actually capture this variation in on the imaging that we do. Next slide please. So I I won't. This is not an image processing talk, so I won't go into detail with this. But I do at this point. I want to give a shout out to Riley and Peter Olver, I think, who are in the audience today. But Riley started in our lab as an undergraduate, but now he's a graduate student in the math department. So he helped us build a pipeline to automate measurements of many of these traits. So we did both manual measurements as well as automated measurements. So not all traits have been automated, measurements have been automated, but some of them are. So very briefly, we kind of, the image processing steps for the different traits that we have, it the basic principles are somewhat the same. Okay, you have like image enhancements, image segmentation, recognition of what trait that you want to measure, and then finally measuring the trait of interest. So the steps are somewhat similar, but the focus on what we want to accomplish, it changes based on which trait you're measuring and how the images are taken or what the images look like. Next slide, please. So the one I showed was leaf, and in this case, this is branch angle. So how do you measure branch angle? How do you, so a person who's measuring it, so undergraduate students are sitting and measuring this using image J. So you can automatically say what you want, but for computer vision, you have to teach the computer to say which part you should focus on. So we had to come up with ways to do that. In this case, we have like the blue strip of plastic that's held against, but Riley had to come up with a way to distinguish which part of this blue strip should be focused on. So he can't, so in this part, challenging things were to identify like the boundary identification and identifying the triangle of interest, and then figuring out which angle should be measured. So in the next slide, please. And this is petiole length. This was another one that a pipeline that he built. So in this case, figuring out which part is petiole, which part is branch. So things that are attached to the plant is branches. So he had to go do a mass dilation to make sure that branches are not identified as petioles. So, and sometimes we image them in the field. So there is a lot of gunk while you're imaging. So how to differentiate things, objects of interest from just general gunk. So those were some of the challenges that that he faced. So I think the point I'm trying to make is, I really had to build individual pipelines for each of these traits. So different traits need to be imaged differently, and they have to be and the measurements, the automating the measurements require us to build pipelines that have different focus. So I think that's what I wanted to appreciate out of this. And so also each of these then is time consuming because it's not one process. Next, please. So while we did this, we also talked about, can we do like a 3D sort of reconstruction and data extraction from that? So instead of just imaging, maybe we can do 3D instead of a 2D, so we can capture other traits that are not amenable to a 2D imaging. So if you want to click on the not that one before, there's a video of this, yeah, on no.
Oh, hearing me, yeah, there we go.
So this is, we took a time-lapse series of a defoliated stem segment, which is placed on our turntable. And so what Riley did then was take individual images of these, and then he did a 3D reconstruction of these images. So this is really very, very preliminary, and we want to invest more time and effort towards it. But I think what we want to do is, for the branch angle, we had to take individual images of each single stem segment and then measure the angle. But what we want to do see is if we can cut down on that and measure branch angle from this 3D reconstruction. If there's if we want to find ways to automatically extract that data out of a 3D reconstructed product. We want to look at how the branches are oriented, the distance between branches, the width of the stem, things like that. These are really hard to do when you do doing like a 2D imaging process. Next, please. But this is still very, very preliminary. And so this has been our workflow for our imaging and image processing. I won't go through this, of course, but if you want to click, Aaron, I want to show you that just the image analysis and data extraction, you manually, when you do it, takes like 12 to 16 weeks. That's like 3 to 4 months. And so when Riley came up with his automated process, essentially it kind of cut down on the time a lot, like 10, just the in the data extraction 10 to 14 days. This is I've not updated these numbers. I don't know what Riley's latest numbers are, but essentially it cuts down on time factor by a lot. If you want to go to the next click, please. Yeah. So the other thing I want to show you is how long does it take to actually image the plants. It takes 6 to 8 weeks. This includes you have to image all of these traits individually in a way that then can go through our image processing pipeline. So that takes a lot of time. So again, I want you to appreciate the time component of this whole thing. And so this is the reason why we can only do one time point and we can't do more than one time point. And that's a problem because we want to see how plants grow during the entire, it takes about 4 to 5 months. And so it just, if you want to do a lot more, you need to automate the process, and so that's the reason we're talking about to you guys, of course. But anyway, so it takes a lot of time though our current process. So next slide, please. And in-field imaging is quite challenging. So I just have a few pictures here. I didn't have a lot of pictures of us in the field, but maybe this is for next year to take pictures. But I want you to see the terrain. This is the cleanest terrain I could find. But usually it's a lot more muddy and more uneven, and there are sometimes weeds on the thing, so you can't quite image plants very well. And as the plant grows, you can see the canopy starts covering, and by the time you reach the third canopy stage, I have here on the left bottom corner, you can see it's completely covered on top. So for somebody to crawl under this and take images, it's really, really hard. And you can see me doing that in the field. We were trying to image branch angle at an earlier time point in the growing season, and it was not fun. And you can see that on the right side, you just, with sitting and defoliating plants, it's kind of fun day for some of the students to kind of spend outside, but it's also slow, and it's a blue sky sunny day, but it's not always like that. It is, some days it's cloudy. One time we had our tents flying out into Fairview Avenue, which is right next to the field, so it was not really good, but it is very, very challenging. So next slide, please. So some people have, there are people who are very interested in this, in understanding canopy characteristics. So LamnaTech has, and so people have built robots for this. There are these field size humongous robots that's there on the leftmost one. That's the LamnaTech field scanalizer. There are more intermediate robots that were built. So it's this one I've shown you is called Phenomobile from CISRO in Australia. And the last one I wanted to show you is this little tiny robot. It's called Terrasentia from over at the University of Illinois. So you have these automated processes, but for the most part, like at least the first two, what it captures is everything from the top view. So you don't see as much canopy from under. For soybean, this is critical because after a certain point, the entire canopy covers and you can't see what's going on underneath. You can't see the branch, especially like the branching features, you can't. So our hope is to find ways to circumvent this problem. I know there has been a lot of progress in robotics now. So we want to go smaller and probably, think of other unique solutions to this sort of our problem. So usually, people utilize what they have and they fit the problem to the existing technology. But what we want is to make or create technology that can fit our problem and not the other way around. So we want a way to customize. We want a way to have a robot that is more customized to our specification, that's easy to use, can be deployed in agricultural setting, which is and also upgradable so things change. So we want it to be more modular. So next slide, please. So I don't know if I didn't want to jump in here, but at least to me, the short-term plan for both image processing and analysis as well as imaging is to build a platform. I'm calling platform kind of loosely. I'm not sure what shape or form it will take, but we want to invest in building a platform to accurately measure the light environment. Again, I call it light environment, not light quantity, but also the quality, how the light scatters within the canopy. So to study that in a larger scale than what we're doing right now. And also we started good with our image processing, but also we want to expand it and improve our existing pipeline for automating data extraction. Our long-term goal, I would say, is that we want to build collaborations that are right now it's us because we know what we want. We know this is an unexplored sort of, it's kind of exciting because it's unexplored. Not a lot of people do what we're doing. Um, and so, but we know we can't do this by ourselves. So we want larger collaboration between people who have the capacity, um, who have the means to make our, what we are thinking into reality. So, we've already collaborated with the math department. And we're talking to you guys. So we want to have longer lasting collaborations with our people within our university, and hopefully with other experts outside the university, to do what we're doing in smaller scale to like a much larger scale. So that's our future plans. So that's it from me.
Thank you very much.
You want
to?
I just wanted to briefly acknowledge. I thought like I was going to do it, but I can do it as well. So right now, the entire shoot architecture project is a collaboration between the Lorenz lab, MnRI lab, and Stupor lab. And interestingly enough, I've been a part of all three labs in the past 6 years. It's been great. So I kind of can speak for all of them and how much interested we are in any further collaborations. We also collaborated with, like I said, with the math department at University of Minnesota, as well as University of St. Thomas. Riley was at University of St. Thomas, and Fumiaki Katagiri, who helped a lot in terms of analyzing some of our data, who's at the CBS. He's a faculty at CBS.
Yeah, I thought you were best to do the acknowledgement, Suma, because you've been really instrumental in putting these labs together and also building the collaboration with mathematics. And so I thought it was appropriate that you did the acknowledgements. So thank you. So now we're ready, Nikos, for questions.
Thank you very much. Questions? So let me ask you, go ahead.
So I have a question about the robot that you said that it goes around the rows. What kind of sensing are you looking for in the field? Are you looking at light sensing, or are you looking at the canopy density? Can you elaborate a little bit about what are you doing from the imaging and sensing in the field?
Sure. I take that question. So what we have done so far is very, very preliminary. Like I said, we only looked at light quantity at any point that we held the line sensor to. But what we want to do is to have an understanding of how the light is scattered within the canopy. So I'll give you an example. So, I don't remember which lab it is, but these guys used a LiDAR system mounted on a robot similar to what I showed, kind of to look at what the under the canopy environment looks like, and to look at how light is scattered. So that's something that we've thought about. That would be cool. So I hope I'm explaining this properly, but we're interested to know how, because the architecture of the plant changes. That means every organ of the plant is placed differently in different genotypes. So we want to know, when that happens, how the light is scattered within the canopy. So how each of the point within the canopy, what kind of light environment it's sensing. So that's, in terms of light, that's what we want to do. But in terms of other traits, we want to see if we can go below the canopy and actually image the plants in a way that we could probably do like a 3D reconstruction of it eventually, and to extract those specific traits that I talked about. So again, this is still very preliminary and things that I've, just something that we've started talking about. So does that answer your question? So I can tell you like more specifically what we want, but that would probably take a while.
No, that's fine, because the thoughts are you guys the goal, is it to do some type of a simulation with ray tracing to kind of understand? I mean, basically, you can simulate a plant that is growing with the number of nodes, and then see what would be the optimal way to capture light on this plant somewhat. Is that the goal from a simulation perspective?
Well, I think there's some people who do something like that, for example, the University of Illinois does a lot of plant simulation and canopy simulation, looking at the distribution of light in simulated situations. But we like to combine that kind of modeling with actual measurements in the field. So actual real ground truth, not ground truth, but real measurements out in the field to get a sense of how actual soybean plants are growing and the variation among different types of soybean varieties.
Right.
Okay.
And
Yeah, that's okay. Thank you.
Yeah, and just to add to that, I think you can, simulation is great, but we also want to know what the basic principles are in order to actually, for us to build a model that is even remotely close to what exists. We need to have an understanding of what the basic units are, how they are built. So I think we won't have that understanding.
Yes, absolutely. I think to kind of prove that your model is right from the simulation, you need to make sure that it corresponds to the right ground truth that you are capturing from the field, right?
Right.
Yep.
Thanks.
Yep. Looking at the effect of, we'd like to know the effect of different shoot architectural properties that may be able to be modified genetically on how they scatter the light and intercept the light in a whole canopy context.
Yes.
So may I ask you a question? So if I understand correctly, and please tell me if I'm wrong, you believe that soybeans are going to be trickier than corn, am I right?
Yeah, I think so, just because of the density of the canopy. There's just a lot of physical interference there going on compared to a corn canopy. Yeah. It's just harder to, like, if you look above a corn canopy, for example, I think you need a lot of information just because it's more open. But a soybean canopy, when it's completely closed, is really closed. It's hard to see inside of it from above. And so you'd have to really kind of go from below, or, we've tossed around crazy ideas like maybe spraying some sort of defoliant on the plant so they all drop their leaves. We're not sure if that'll work, but ideally that would be the best scenario. We go out there and spray a defoliant, they'd all drop their leaves, and then we have all the plant skeletons out there. We can image those. And
Aaron, that's an idea I tried out 1 year. So it was not great because the chemical that I used was Roundup. So it essentially kind of shrunk the plant a little bit.
Yeah.
So what did not, I think that's a worthwhile idea, mostly because then we have to play with different kind of sprays. I just tried one. So we want to spray that will only defoliate without changing the architecture. So it's not an idea without merit, but that's something that is a possibility. Then we won't have the leaves, and so.
Yeah. So that's one. I don't know if that'll work, but that other things we talked about. Well, of course, somehow looking underneath the plants is a possibility. We've also just kind of tossed around the idea of not even really getting to the individual architectural features like branch angles and number of branches and stuff like that, and rather use something like LiDAR to get a sense of how much light may transmit into the canopy. It's sort of a surrogate for what that canopy may look like underneath.
Are you extracting some features from the plant or from the field, like the number, the density of the plant, or the leaf sizes, or things like that can try to forecast your yield?
No, we're not. not really trying to forecast yield. I guess we're sort of forecasting yield in a way. I mean, we're trying to figure out what about those individual plant features like leaf size or branch number, for example, correspond to different favorable properties for the whole canopy at the field scale, and then figure out what of those properties would lend themselves to maybe a higher yield potential for breeding plants that had a better canopy. But in terms of just overall yield estimation, for example, mid-season, it's really hard to do that in soybean from kind of just a sensing standpoint. I don't think much of that has been done. You can use basic sensors and calculate things like what we call vegetation indices, NDVI, stuff like that, and that'll kind of give you an idea of what yield may be in one part of the field versus another part of the field, or maybe yield might be in one county versus another county. It'll give you a ballpark estimation. But if a farmer really wants to forecast what yield he may expect, right now he kind of has to go out there and just sort of pull plants and count pods and seeds and stuff like that, kind of mid-season. So.
But I think the principle is that the architecture affects yield, and we've seen that in other plants. So we want to do those feature extraction and hopefully tie that to yield.
But you could, I mean, if you could, for example, if you think about a technology that would maybe allow you to drive a robot, for example, under the plants in the row, and then sense different plant architectural features, at a certain time of year, the stems contain pods. And so maybe you could use the same technology to actually drive through the field under the row and get an estimation of how many pods are on the plants. And that could be used for kind of yield forecasting. And that'd be very valuable for, organizations that are in the business of yield forecasting. USDA, for example, does yield estimates in certain times of the year.
Yes, I was thinking about a combination of a drone and, looking at the macro from the top and then looking at certain places with the robot.
Yeah.
But the whole idea is about that. What do you want to look at? What features do you want to extract in a way to kind of start predicting yield? And then ultimately, I think what you want is which plant is best for the right location in the Midwest or up north or down south or something like that, right?
Yeah, I mean, so there's a difference between what we may want from as geneticists compared to someone who may work for the USDA, for example, who wants to forecast yield for market reasons. So we as geneticists want to understand ways that we can modify that crop canopy for the potential to increase overall yield, in the longer term. So understanding how we may design a canopy basically that will increase yield.
Yeah. Yes.
So one thing I also want to mention, and is the decades study we are doing. So we have this little study that we are looking at how the architecture has changed over the last 6 or 7 decades. And we hope to see if some of the features that we are studying of shoot architecture has changed and yield has kept in touch with that. So I know correlation is not causation, but we want to see how any of these features, how does it correlate over time with increase in yield that we've seen in the past decade. So this kind of speaks to your forecasting, not exactly, but to some extent.
Hi, I have a question. Interesting talk. So did you present something, I didn't see it, that showed kind of the early genesis when the plant is first growing, what the architecture generally might look like? So the reason one thing, just bringing up, practically speaking, if you're able to measure some of the earlier features when the plant is smaller, how well that might correlate to when the plant is much larger and obviously a more complex problem to, measure the solar radiation, light penetration, all the other things that you want.
Yeah, that's a good point. I don't think, unfortunately, I mean, a lot of that canopy structure is determined by branch angle, orientation, number, things like that. And those branches don't really come out, well, I guess, come out a little bit earlier. So assume you've done a lot of this measurement. How much does the branch properties correspond from early development towards later development?
Yeah. So some of the features don't change a lot, like branch angle doesn't change a whole lot. So I want to, I think I kind of said that during the talk, that with the current process we have, we can only afford to do it at one time point. So we chose a time point when we know from other studies that maximum photosynthesis occurs, and that's at the time when the canopy is completely covered. But your point is absolutely spot on. So it would be amazing to do at least a few time points and see which of these features actually change over time and which of them don't. Maybe we have to focus on things that don't actually change and maybe capture those when plants are more amenable to
Yeah.
robotic extraction.
Yeah, right. You said it. Yeah, exactly. And because, and once you find those, I don't know exactly what that'd be. I'm sort of imagining in my mind, the speed up animation when you see plants grow, you've seen a million times, and you kind of can almost see these patterns.
Right.
But if there's something that's prevalent, then you might be able to sample earlier and more quickly with a robotic platform and an earlier stage that could do it where you can't do that now.
Yeah, absolutely.
I mean, it's obviously one question has to be answered before you could design the right robot, but it could simplify and diversify the design of a robot then.
Yeah.
That's my only,
Yeah, we'd look into that. Soybean plants are very plastic. So, the obvious thing we'd like to do the most is grow things in controlled conditions and greenhouses and stuff like that, do these things. But for these traits in soybean, that's not a possibility at all. They change so much in the field. So
I think what is possible is that we select a few maybe genotypes and then kind of do the same sort of process we've done with 40 genotypes, but maybe do it with like 6 or 7, and then track all these features at multiple developmental stages and see which of them change and which of them don't a lot. Maybe that's one way to do it. It's just, constrained by how much we can do.
Any other comments, questions? So thank you very much, Aaron and Suma. Have a great weekend.